Hi viewers, this is your Raju sir. Today we shall discuss about classification of bacteria. So we are showing on the screen classification of the bacteria. So my dear students, we must be reading to this classification of the bacteria. It is very easy and it is very useful. So first of all, therefore, here the classification of the bacteria. Bacteria can be classified in many ways. The first classification scheme was published in 1773 and more have appeared since. Science of microbiology has developed on the kinds of classification, but medically important classification is as well follow. So we are observing here the classification is 1773 first classified to the bacteria. So here the bacterial classification based on the many ways are there. So let I will discuss one by one. So we are showing about to the screen. So first one, my dear students. So based on the several major properties. So first of all, we are based on the several major properties by the using to classify to the bacteria. So first one, bacteria, morphological major factor. So this is morphological major factor, and anatomical major factor, and staining major factor, and based on the pathogenic major factor, and based on the relationship of the host and the organism, and nutrition and environmental factors. So those are involving to classify to the bacteria. Keep it your mind. And next we are learning about the first one, morphological major factor. So here the morphological major factor. The bacteria can be classified according to the morphological characteristics. So like their cell shape and structure and their specific arrangement, like mostly motile and flagella arrangement. So bacteria can be classified into the 6 major groups on the morphological basis. So first one major group, true bacteria. So first one bacteria here, the bacteria is having the major group, six major groups. So in this 6 major groups, first one true bacteria. So we shall show to this image true bacteria. True bacteria. Here the true bacteria is having the Coccus bacteria. So these are spherical or oval shape of the bacteria. So this one bacteria we are observing this Coccus bacteria. So this one bacteria is spherical or oval shape of the bacteria. So here the bacteria arrangement of the individual's organism can be classified, can be described as, so first one Monococcus, second one Diplococcus, and third one Streptococcus, and fourth one Streptococcus, Tetracoccus, and such. Here the 6 groups is containing to the bacteria. Coccus bacteria is having 1, 2, 3, 4, 5, 6 and 7. So 7 is containing to the bacteria, this one Coccus bacteria. So first one go Monococcus bacteria. Here the Monococcus bacteria is having the single spherical structure of the bacteria. So this one Monococcus bacteria. So we are easily observing. Next one a Diplococcus bacteria and a Streptococcus bacteria, Streptococcus pneumonia. So this is an example of the Streptococcus bacteria. So here the Streptococcus bacteria is having the pairs of the individuals, and Streptococcus and the Streptococcus tetra such. So we are observing to this bacteria, and we are show this bacteria, and another one Bacillus Bacteries. So first of all, my dear students, we are observing to this bacteria. So we are showing to this image. So here the image is containing to the types of the bacteria. So first one bacteria we are observing to here. So this one single spherical structure of the bacteria is also called as Monococcus bacteria, and here the structure we are observing to this structure. So here the double structure of this helical structure of the bacteria is also called as Diplococcus bacteria. So here the structure, so we are observing to the structure. So here the structure is containing to the number of the cocci. So this one bacteria is also called as Staphylococcus bacteria. So here the bacteria, here the chain structure of the bacteria. So cell division on one place, in one place of the bacteria. So this one single cell, so single cell is divisible to form to the two cells. This is a Diplococcus. So this is Monococcus, Diplococcus, Staphylococcus. We are observing to this bacteria. And the next one here observing to this pocket structure of the bacteria. So here the pocket structure of the bacteria मतलब क्या करना, cell division into the more plants perpendicular to one to another. For example, we are observing to this bacterium. So this bacteria is this bacteria we are observing to this bacterium. So here the bacteria is divisible to form to this type of the bacteria. So this type of the bacteria is also called which type of the bacteria. So here the bacteria we are observing to this bacteria. So first one देखो, so Monococcus bacterium, Monococcus species में. So Diplococcus bacteria, pairs of the bacteria, Streptococcus pneumonia, and Staphylococcus bacteria is also Staphylococcus aureus, and the next one देखो, Streptococcus bacteria, Streptococcus प्रोजनी में वो स्ट्रेप्टोकोकस बैक्टीरिया. और क्या है नेक्स्ट वन देखो, टेट्राकोकस बैक्टीरिया. टेट्राकोकस बैक्टीरिया ग्रुप ऑफ द फोर बैक्टीरिया. सो ग्रुप ऑफ द फोर बैक्टीरिया माइक्रोकोकस स्पीसीज में सार्सना बैक्टीरिया इज ऑल्सो हैव ऑन द नंबर ऑफ द बैक्टीरिया वी आर ऑब्जर्विंग. सो इन दिस बैक्टीरिया में आप लोग देखिए फर्स्ट ऑफ ऑल स्ट्रेप्टोकोकस बैक्टीरिया कैसा होगा देखो. सो आप लोग हम देखिए, सो दिस इज ए मोनोकोकस, this is a monococcus, and this is a diplococcus, this is a staphylococcus, and this one streptococcus, this one pocket shape of the bacteria, and this one देखो another one देखिए, this one another one देखो, यहाँ पर देखिए. सो क्लस्टर डिवीजन ऑफ द बैक्टीरिया यहाँ पर देखो, दिस इज ए सार्सना बैक्टीरिया एंड टेट्राकोकस बैक्टीरिया यहाँ पर देखिए, दिस इज ए टेट्राकोकस. सारे बैक्टीरिया हैव इन द बैक्टीरिया टेट्राकोकस बैक्टीरिया. आप लोग सार्सना बैक्टीरिया ऑब्जर्व करिए. सो दैट इज ए बैक्टीरिया. वो स्ट्रक्चर के लिए आप लोगों को वो स्ट्रक्चर करना चाहिए, कहाँ पर वो नोट में करो. एंड नेक्स्ट वन देखो, लेट वी शैल डिस्कस अबाउट दिस बैक्टीरिया. So this one देखिए, this one is we are observing to this bacterial shape. First one coccus bacteria. So coccus bacteria, this is a monococcus bacteria. This is a monococcus bacteria. अरे बाप. So this one monococcus bacteria. And this one Diplococcus bacteria. So Monococcus, Diplococcus bacteria. And this one देखो, so here the Diplococcus bacteria is converted to the Staphylococcus bacteria. So here the Staphylococcus bacteria के बाद Streptococcus bacteria. This one देखो, this is a Streptococcus bacteria. Streptococcus bacteria के बाद क्या करना, सार्सना बैक्टीरिया आप लोगों में ऑब्जर्व करना चाहिए. So this one बैक्टीरिया के लिए, कोकस बैक्टीरिया के लिए, कोकी बैक्टीरिया के लिए आप लोग वो डायग्राम्स करो. एंड नेक्स्ट वन देखिए, बैसिलस बैक्टीरिया. सो बैसिलस बैक्टीरिया इज ऑल्सो वोवल शेप ऑफ द बैक्टीरिया और रॉड शेप ऑफ द बैक्टीरिया. सो वोवल शेप और रॉड शेप ऑफ द बैक्टीरिया. दिस वन स्पेरिकल शेप ऑफ द बैक्टीरिया और वोवल शेप ऑफ द बैक्टीरिया. सो दिस वन रॉड शेप ऑफ द बैक्टीरिया, बैसिलस बैक्टीरिया. सो यार द बैसिलस बैक्टीरिया में क्या करना ना, कोकोबैसिलस फर्स्ट वन है. सो दिस इज ए मोनोबैसिलस बोलते हैं दिस वन का. And the second one can here देखो Bacillus. So this one Monobacillus. So here the Bacillus में क्या करना, Diplobacillus. Same to same वो bacteria same to same side by side having this bacteria. And the next one क्या करना ना, Streptobacillus. Bacillus 1, 2, 3, 4. So 4 is containing to this bacteria Bacillus. And third one देखिए पैलिसाइड बैसिलस. पैलिसाइड मतलब क्या करना, वो सेल्स देखो ऊपर से ना पैलिसाइड टिश्यू बोलते हैं ना. So पैलिसाइड टिश्यू is containing to the which one पैलिसाइड टिश्यू have in the which type of the arrangement, pillar arrangement, like about here the bacterial arrangement is also पैलिसाइड arrangement. So पैलिसाइड बैक्टीरिया दिस वन आप लोग ऑब्जर्व करिए. एंड नेक्स्ट वन देखो वो नीचे है, वो नीचे क्या करना ना, नेक्स्ट वन बैक्टीरिया विब्रियो बैक्टीरिया. So let we'll discuss about this bacteria. So वो विब्रियो बैक्टीरिया में देखो दिस वन ट्रिप्टा एंड स्टाल्क एंड दो आप लोग ऑब्जर्व करिए. So this one के लिए वो विब्रियो, दिस वन कामा शेप ऑफ द बैक्टीरिया. सो दिस वन क्या कर रहे हैं, दिस वन आप लोगों को पता है वो टेड शेप ऑफ द बैक्टीरिया. दिस वन कामा शेप ऑफ द बैक्टीरिया आप लोग अंडरस्टैंडिंग करना चाहिए. वो इमेज आप लोगों को करना चाहिए, दिस वन इमेजेस मस्ट भी करना चाहिए. सो नेक्स्ट वन देखिए हियर द. सो दिस वन बैक्टीरिया में देखो. सो यहाँ पर देखिए. सो बैसिलस कितना टाइप, देर आर द रॉड शेप्ड बैक्टीरियाज ऑन द बेसिस ऑफ द अरेंजमेंट ऑर्गेनिज्म. दे कैन बी डिस्क्राइब एज डिप्लोब्लासिलस, स्टिप्लोबैसिलस, पैलिसाइड एंड चेन लेटर फॉर्मेशन एंड कोकोबैसिलस एंड कॉमन शेप्ड बैसिलस. ऑलरेडी आप लोगों को वो इमेज में दिख रहा है, वो बैसिलस के लिए आप लोग देखो वो इमेज में दिख रहा है. सो दिस वन देखो कोकोबैसिलस एंड बैसिलस एंड डिप्लोब्लासिलस, स्ट्रेप्टोबैसिलस, पैलिसाइड बैसिलस आप लोगों को वो इमेज दिख रहा है. सो दैट्स व्हाई वी आर ऑब्जर्विंग हियर द बैसिलस बैक्टीरिया डिपेंड अपॉन द शेप द बैक्टीरियल क्लासिफिकेशन. एंड नेक्स्ट वन देखो. सो मॉर्फोलॉजिकल क्लासिफिकेशन ऑफ द बैक्टीरिया. सो यहाँ पर क्या कर रहे ना, सो हियर द बैसिलस के बाद देखो, बैसिलस के बाद क्या क्या होगा, लेट आई विल शो टू दिस बैक्टीरिया. सो कीप इट योर माइंड माय डियर स्टूडेंट्स. नेक्स्ट वन वी आर ऑब्जर्विंग टू दिस actinomycetes. Actin ray makes a figures. We are observing here the actinomycetes. These are rigid organism like a true bacteria, but they resemble fungi in they exhibit to the branches and tend to the form of the filaments. So आप लोग वो आइडेंटिफाई करिए, वो इमेज आइडेंटिफाई कर रहे तो आप लोग क्या अंडरस्टैंडिंग हो गया ना. यहाँ पर वो इमेज आइडेंटिफाई करेगा तो सो फंगेनल ऑर्गेनिज्म वो आइडेंटिफाई होगा, वो इमेज आइडेंटिफाई करेगा तो ये ऑर्गेनिज्म फंगेनल ऑर्गेनिज्म. सो फंगेनल ऑर्गेनिज्म सेम टू सेम है. सो इसलिए क्या बोलते हैं एक्टिनोमाइसिटिस. माइसिटिस मतलब फंगेनल ऑर्गन. So here the fungal organ like that, here the actin race is having the bacteria. So like about the actinomycetes bacteria we are observing to this one. And the next one Spirocities. These are relatively longer, slender, non-branched microorganisms. So this is a spiral shape of the having the several colis. So this one देखो. So this one image spiral shape of this image. So this one bacteria is or spiral shape of the bacteria के लिए और next one देखिए mycoplasma. So mycoplasma bacteria है. So mycoplasma these bacteria is lack in the rigid cell wall. So वो bacteria में क्या करना है, cell wall नहीं है. And next one क्या करना है, and they highly pleomorphic and have identified the shape, pleomorphic shape. They occurs into the round and oval body shape of this bacteria. So this one bacteria shape. And rickets and chlamydia. So rickets and chlamydia आप लोगों को पता है, these are very small and obligatory parasites. Rickets and chlamydia. So and one time of very considered closely related to the various now these are the recorded as the bacteria. This one रिकेट बैक्टीरिया के लिए आप लोगों को वो देखना चाहिए. And the next one structure based on the anatomical structure or anatomical features of the bacteria. So my dear students, we are observing to this anatomical structure of the bacteria. In this anatomical structure, you know that the capsulate and the non-capsulate of the bacteria based on the anatomical structure. We are observing to this anatomical structure. So here they go this one structure. So this is a capsule structure. So this one outer layer is called capsule structure. We are shown to this color tree. So outer layer of this structure is called capsule structure, and inside having the structure is called cell wall. This one. So this yellow color cell wall. So this one light green color outer layer capsule structure. So but some of the bacteria and otherwise they go this one plasma membrane layer. So plasma membrane layer inside is having the cytoplasm and DNA. So आप लोग internal structure easy understanding करना चाहिए. And here the bacteria we are observing to this bacteria. So here the bacteria based on this anatomical structure, capsule bacteria is having two types, capsulated and non-capsulated. So some of the bacteria that about this Streptococcus pneumoniae bacteria is containing the capsule structure, but non-capsulated bacteria are there. Viridans Streptococcus. Viridans मतलब like about having the small symptoms of the viruses is also containing to this bacteria. So viridans Streptococcus bacteria couldn't have into the capsulator. So आप लोगों को understanding करेंगे. And the next one here the depend upon the flagella. Depend upon the flagella, bacteria is also classified into some types. So first one here the flagellate of the bacteria. So here the flagellate is having the four types of the flagellate. First one monotrichous, lopotrichous, amphitherotrichous, petrotrichous. Aflagellated bacteria. So aflagellated मतलब couldn't have into the any flagellated. So here this is a monotrichous. Here the bacteria is containing to the only one flagella. Such a kinds of the bacteria is called monotrichous bacteria. So bacteria is containing to four flagellated. So this one. So bacteria is each flagellated is equal of the flagellated. That bacteria is called Lopotrichous bacteria. So this one. So here the bacteria is containing to the both flagellated, both side poles. So that matlab this is a pole and this is a pole. So this pole bacteria is containing to the flagellated. So that bacteria is called amphitrichous bacteria. And another one last one peritrichous bacteria. Here the bacteria is containing to the more than flagellated. Such a kinds of the bacteria are called peritrichous bacteria. So we are easily observing to this bacteria. So this is a peritrichous bacteria. So another one non-flagellated bacteria. Here some bacteria is an example of the Shigella species. Shigella species is an example of the non-flagellated bacteria. So here the non-flagellated bacteria couldn't have into the any flagella. So what has to this flagellated? Flagellated it used to move to the one place to another place. That's why here they use it to the flagellated. Flagellated is one of the locomotor of the bacteria. So next one they go pores. So classification to the pores of the bacteria. So here the spores is forming to the Bacillus species. So Bacillus species is formation to the number of the spores. And another one non-sporing bacteria, Escherichia coli. Escherichia coli is good into formation to the flagellate spores. So we are learning to this one. And the next one they come. So next one based on the staining reaction. So here the bacteria is two types of the bacteria. So first gram staining bacteria and acid fast bacteria. Acid fast bacteria, gram staining bacteria. So first one here the gram staining bacteria. We are observing to the gram staining bacteria. So here the gram staining bacteria is two types of the bacteria, gram positive and gram negative bacteria. So here this one they go after the gram staining bacteria organism which occurs into the violet in color. So gram staining bacteria is obtained to the violet color. आप लोगों को पता है ग्राम नेगेटिव बैक्टीरिया appears to the pink color or red color. So ग्राम नेगेटिव में पिंक और रेड, ग्राम स्ट्रेनिंग में क्या करना वायलेट एंड red. That's why here the bacteria is two types of the bacteria based on the types. And another one देखो, so another one देखो acidic faster strain of the bacteria. Acidic faster organisms after the Nielsen strains, it will show a pink color, but non-acidic bacteria are there, it will show a pink color. So, but non-acidic faster organisms after the strain organism will appear to the blue in color. So some bacteria क्या करना है, some bacteria में क्या करना है, जेल नील स्ट्रेन करेगा तो क्या करना है पिंक कलर होता है, और some bacteria में क्या कर रहा है ना appears to the blue color. So we are observing to this acid faster strain, gram strain. So keep it your mind, must be observed here. And next one देखो gram staining bacteria में देखिए. So gram staining bacteria, gram positive coccus bacteria, Staphylococcus pneumoniae, and gram staining bacteria में आप लोग हम देखिए दिस वन gram staining bacteria में. So Staphylococcus pneumoniae, Staphylococcus aureus, and gram negative coccus Neisseria gonorrhea. So this one gonorrhea disease के लिए वो बैक्टीरिया है. Staphylococcus aureus is caused to the Streptococcus pneumonia disease के लिए. And gram positive bacteria, rod shape of the bacteria, this is a Clostridium bacteria. And gram negative bacteria में क्या करना rod shaped bacteria, E. coli bacteria, those are the stain method of the bacteria. So stain method में gram positive कैसे आइडेंटिफाई कर रहे हैं, पिंक कलर कलर obtain करेगा तो क्या करना gram positive है. So और another one कलर क्या करना red or blue color obtain करेगा तो that one gram negative, pink or blue color करेगा तो so this one gram positive negative bacteria बोलते हैं. And acidic antibacteria, an example of the Mycobacterium tuberculosis, it is very very important. Acid fast Bacillus bacteria and non-acid Bacillus Staphylococcus aureus. This one is also Staphylococcus aureus. So we are example this one. And the next one देखो. So next one based on the pathogenetic classification. Based on the pathogenetic classification. So pathogens, the organism which is able to spread disease. The organism which is able to spread disease can cause us to the diseases. So spread to the diseases can cause us to the diseases. So that organism is called pathogenic organism. And a non-pathogenic organism which does not cause us any diseases. For example, lactobacillus. So lactobacillus is a non-pathogenic organism. In our digestive system is also containing to the one of the bacteria, E. coli bacteria. E. coli bacteria is a non-pathogenic organism. So that is not harmful in our body. So our body में क्या करना harmful नहीं है, E. coli bacteria understanding करिए. So next one करना common cells. So normally non-pathogenic, but sometimes they show disease when this immediately impaired. So some bacteria क्या करना actually non-pathogenic bacteria है, but sometimes क्या करना it will cause us to the diseases. Such as bacteria is called common cell bacteria. So we are observing this one. And the next one देखो based on the relationship of the host and the organism. So here the classified to the bacteria. So first one is saprophyte bacteria and parasite bacteria and common cells of the bacteria. Three types of the bacteria is classified to the here. So first one क्या करना saprophytic bacteria, free living organism or microbes, dead animals में क्या कर रहे हैं ना वो saprophytic bacteria always take to the food material, dead organism material. And the next one parasitic bacteria क्या करना that establish themselves and multiple in the host. Host में क्या करना first जाना के बाद क्या करना ना so उनका organism का group क्या करना ना establish कर रहा है. So that one आप लोग में identify करिए. And the next one common cells, microbes that lives in the completely live the harmony but they cannot produce diseases. Diseases when the host resistance is the low. For example normal flora organisms example of this bacteria आप लोग में identify करिए easy होगा. और नेक्स्ट वन क्या कर रहा ना नेक्स्ट वन So next one देखिए, next one. So next one देखो, based on the nutrition. Based on the nutrition, here the bacteria is classified to the two types of the bacteria. So first one, autotrophous bacteria and heterotrophous bacteria. So here the that my dear students, here the some of the bacteria is bacteria prefer to the own food material. Some bacteria is prefer to the own food material. So such a kinds of the bacteria is called autotrophic bacteria. And some bacteria is couldn't prepare to the own food material. It is always based on the other organism in the body. That type of the bacteria is called heterotrophous bacteria. Heterotrophous bacteria. So we must be observing to this here. So this one observe करना चाहिए. And the next one देखो. And the next one, temperature. Based on the temperature, the bacteria is classified to the four types. So first one, Phycophilus and Mesophilus and thermophilic and extremely thermophilic. So here the bacteria are there. First one, Phycophilic bacteria. So this one, Phycophilic bacteria. This one bacteria is lived under the 15 to 20 degrees temperature. Example, Pseudomonas flunaria, fluorescency. Fluorescency. So Pseudomonas fluorescency, it is very example of that. Example of the Psychophilus bacteria, and another one Mesophilus bacteria. So this one lived under the 20 to 40 degrees temperature. 20 to 40 degrees temperature. An example of the Escherichia coli. इसलिए क्या करना, वो Escherichia coli ना हमारा डाइजेस्टिव सिस्टम में वो लिविंग कर सकते हैं. बिकॉज क्या करना ना, हमारा बॉडी का टेम्परेचर कितना होगा ना, 37 डिग्रीज टेम्परेचर है. 37 डिग्रीज मतलब 36 टू 37 डिग्रीज टेम्परेचर. इसलिए क्या करना, Escherichia coli ना लिविंग कर सकते हैं, हम लोगों को यूज़ कर सकते हैं, हम लोगों को यूजफुल कर सकते हैं. एंड नेक्स्ट वन क्या करना, Salmonella enteria. And the next one, Streptococcus aureus. So those are the bacteria, an example of the Mesophilus bacteria. And the next one, Thermophilus bacteria. You know that my dear students, thermo मतलब temperature, high temperature में low temperature में क्या करना, वो बैक्टीरिया लिविंग कर सकते हैं. दैट मतलब वो 50 to 60 डिग्री टेम्परेचर में क्या करना, वो लिविंग कर सकते हैं. एन एग्जाम्पल ऑफ Bacillus and another one, Bacillus stereothermic bacteria is an example of them. An example of the thermophilic bacteria. You know that here the temperature 50 to 60 degrees temperature का ऊपर मिलता है ना. So in this 50 to 60 degrees temperature मतलब क्या करना, राजस्थान जैसलमेर में वो क्या करना, डिजास्टर में वो इतना टेम्परेचर मिलता है. सो इस तरफ आदमी को वो पर से क्या करना, वो बैक्टीरिया लिविंग कर सकते हैं. बेकार क्या करना, दिस वन बैक्टीरिया से thermophilic bacteria है. सो नेक्स्ट वन क्या करना, एक्सट्रीमली हाई टेम्परेचर बैक्टीरिया. सो दिस वन बैक्टीरिया क्या करना, हाई टेम्परेचर में क्या करना, लिविंग कर सकता है. 250 डिग्रीज टेम्परेचर में को लिविंग कर सकता है. सो वो वाला आप लोग अंडरस्टैंडिंग करिए. बैक्टीरिया टेम्परेचर के लिए बैक्टीरिया इज क्लासिफाइड टू द 4 टाइप्स. सो फाइकोथर्मिक एंड मेसोथर्मिक, थर्मोफिलिक एंड एक्सट्रीमली थर्मोफिलिक के लिए आप लोगों को क्लासिफाई करना चाहिए. एंड नेक्स्ट क्या करना, ऑक्सीजन डिपेंडेंट बैक्टीरिया. So oxygen dependent bacteria, my dear students, here the oxygen dependent bacteria is 3 types, classified to the 3 types of the bacteria. So aerobe bacteria, and second one obligative aerobe, and third one micro aerophilic bacteria. So aerobe bacteria मतलब क्या करना, growing in ambient temperature, so which is the consist of the 21% oxygen and a small amount of carbon dioxide 0.3. वो परसेंटेज में क्या कर रहा है ना, वो बैक्टीरिया लिविंग कर सकते हैं. दैट मतलब नेचुरल एनवायरमेंटल में क्या करना, लिविंग कर सकते हैं वो बैक्टीरिया. वो बैक्टीरिया लिविंग कर सकते हैं. एंड नेक्स्ट वन क्या करना, ओब्लिगेटिव एरोब्स. ओब्लिगेटिव एरोब्स, एनारोब्स आप लोगों को पता है ना. ओब्लिगेटिव एरोब्स, सो स्ट्रिक्टली रिक्वायर्ड टू द ऑक्सीजन फॉर देयर ग्रोथ. ऑक्सीजन नहीं तो वो ग्रोथ नहीं है. एग्जाम्पल Pseudomonas aeruginosa, an example of this obligative aerobes. And micro aerophilic, so grow under the reduce oxygen, like about 5 to 10% oxygen, and increasing to the CO2 8 to 10%. यहाँ पर क्या करना, थोड़ा ऑक्सीजन है तो वो ग्रोइंग कर सकते, ज़्यादा ऑक्सीजन है तो क्या करना, वो मर गए वो वाला. सो माइक्रो एरोफिलिक्स वो बैक्टीरिया से एग्जाम्पल क्या करना ना, क्लामिडोबैक्टीरिया जेजुनियम एंड हेलिकोबैक्टीरिया पाइलोरिया. Clamidobacteria jejunium, an example of this bacteria. And next one देखो classification, bacterial classification में. So bacterial classification, pH content of the bacterial classification. So acidophilic lactobacillus an example, alcophilic vibrio, and neutrophilic pH 8 to 9. And the measure of this measure of the medically important to this bacteria. So आप लोगों को आइडेंटिफाई करना चाहिए वो बैक्टीरिया के लिए. Keep it your mind, my dear students. And the last one goes to, so otherwise the classification of the bacteria. So motile and non-motile bacteria, and pathogenic and non-pathogenic bacteria, and sensitive and resistant bacteria, and lactose fumaria bacteria. We are easily give to an example of the bacteria. So those are the bacterial classification. So we are observing to this bacterial classification. So here the bacterial classification is based on to the 6 major types already मैं बोल दिया. So bacterial classification, first one morphological structure, anatomical structure, staining structure based on the pathogenic structure, based on the relationship of the host organism and nutrition and environmental. So those are necessary for learning to the bacterial classification. Thank you.